This is the Dyce Lab Summer 2019 Research Update, and in this brief video, we'll be giving highlights from two different projects. The first is from the New York State Beekeeper Tech Team. This is a program that's funded by the New York State Environmental Protection Fund, as well as partially by donations from the Empire State Honey Producers Association. The second project, we're going to hear from our postdoc, Dr. Christine Urbanowitz, talking about pesticide risk to honeybees. And this is a project that she just published in the May issue of Apidology, and it's one that is also funded by the New York State Environmental Protection Fund, as well as the USDA. So let's start with the tech team. The tech team is a program that started back in 2016 to actually identify what are the major challenges that beekeepers are experiencing here in New York State, and what are some things that we can do to try to improve colony health and reduce colony losses. The data that I'm going to be presenting today are from working with 34 beekeepers back in 2017 and 2018. With these beekeepers, we visit their operations and we sample colonies. And we sampled 320 colonies from this cohort. And when we're sampling colonies, we're collecting a variety of data, including the Varroa mite levels of those colonies, nosema levels, the population of that hive by estimating the frames of bees, and then we also collect samples to look at virus levels. And we're testing for 6 different viruses, the same ones that the National Honeybee Survey are testing for. These are deformed wing virus, acute bee paralysis virus, Israeli acute paralysis virus, chronic bee paralysis virus, Kashmir bee virus, and Lake Sinai virus 2. We then ask beekeepers to keep track of which of the colonies we sampled survived or died over winter. And we actually heard back from a lot of beekeepers, and we were able to track the survivorship from 154 colonies, which is a pretty good sample size for this. And so based on these colonies that are either alive or dead in spring, we can then look back at what we saw in those colonies in fall, in September when we sampled them, and we can see if any of the things we measured are actually predictors of winter mortality. And what we found is that 3 things emerged as being significant predictors of winter mortality here in New York State. The first one probably comes as no surprise, and that is Varroa mites. The higher the levels of Varroa mites in fall, the higher the probability of colony loss over winter. So if we take a look at this graph, we can see along the bottom are our Varroa mite levels in the fall, in September specifically, written in mites per 100 bees. And along the y-axis, we actually see the probability of winter survival. This black line is our average probability of survivorship, and the yellow shaded area is actually our 95% confidence interval. The yellow dots at the top are colonies that survived over winter, and those are the corresponding Varroa mite levels they had in fall. And the dots along the bottom are colonies that died over winter, and their corresponding Varroa mite levels in the fall. So we can see generally the trend is the higher the Varroa mite levels are in September, the lower the probability is that your colony will survive over winter. And if we kind of specifically pull out some numbers here, for instance, we can see that if a beekeeper in September of 2017 did an alcohol wash and found that there were 3 mites per 100 bees in the fall, this is actually the treatment threshold that we recommend here in New York State for beekeepers to put a treatment on if they're at or above 3 mites per 100 bees. So if they find that they have 3 mites per 100 bees in the fall, the average probability of survival over winter is about 50%. Now this gets a lot lower as mite levels get higher. For instance, if you found in September that mite levels were about 11 mites per 100 bees, then the probability of survivorship over winter goes down by about 25%. So it's really critical that beekeepers are actually aware of their mite levels throughout the year and are ensuring that colonies have low levels of mites in the fall when those colonies are preparing for winter. And by doing so, we're giving our colonies the best chance of winter survival. So it was interesting because we also found 2 viruses emerged as significant predictors of winter mortality as well here in New York State. And those viruses were Lake Sinai virus 2 and chronic bee paralysis virus. And it's interesting to me because these viruses actually weren't the most common viruses that we found in colonies here in New York State. They were a little bit more on the rare side. For instance, Lake Sinai virus 2 was found at varying levels in just under 20% of colonies, and only 10% of colonies had any detectable levels of chronic bee paralysis virus. And so some of these colonies still looked very healthy and virus levels were low, and other colonies had very high levels of these viruses. And what our data are showing is that if colonies do have high levels of these 2 viruses, either one of these 2 viruses, then the probability of winter survival is low. We also found some very clear benefits to monitoring. The more often a beekeeper monitors during a year, the lower the Varroa levels are in autumn. It really pays to be aware of their parasite population so you can act accordingly before levels get out of control. So beekeepers who monitored at least 3 times a year had average Varroa levels below the treatment threshold of 3 mites per 100 bees by fall. And beekeepers who monitored even more, say 8 times a year, had levels below 2 mites per 100 bees. We recommend beekeepers monitor monthly as time and labor and weather permits, but to monitor a minimum of 4 times during the year just to be on the safe side. Typically, if you are doing it 4 times a year, the times that you're looking at are as soon as colonies come out of winter on a nice mild day, right before you're putting your honey supers on, right as soon as you're taking your honey supers off, which typically is around August or September for beekeepers here in New York, and then again in October, which is the time when Varroa mites reach their peak here in New York State. We recommend that beekeepers apply a treatment anytime levels reach or exceed that treatment threshold. Also, as no surprise, we saw a similar trend with the number of treatments that beekeepers were giving their colonies. The more treatments a colony received, the lower the Varroa mites were in fall. I give these results cautiously. I am definitely not encouraging beekeepers to apply 10 different treatments to their colonies every single year, but instead to only apply treatments when they need to, when those colonies reach or exceed the treatment threshold. And this is different for every operation, right? It depends on the genetics of those bees, so how resistant are they to Varroa to begin with, and it depends on the environment. For instance, are those colonies in an area with a lot of Varroa pressure from other neighboring hives? Or do they winter in an area where they get a nice brood break for a couple months that can help reduce some of those Varroa levels? And so because all operations are different, they might require different numbers of treatments. So one thing that you can take away from this is to just be prepared. We know that in 2017, beekeepers needed to apply at least 3 treatments in order to see mite levels below 3 mites per 100 bees on average. And so maybe order a few treatments, have them on hand in case you need them. Plan in advance what types of treatments you like to use and kind of what times during the year you typically use them. If you find that you do need to use them that year, then you're ready and you just have them on hand and can put them right in. If you don't need to use them, then that's great. That's something that you can keep around for the next year. But all of this is just kind of showing ways that we can be more informed about our colony health and ways that we can act more accordingly might have benefits in the end. So that is our research update for the tech team. Now let's turn it over to Dr. Christine Urbanowitz, and she will switch gears and talk a little bit more about pesticides.
So beekeepers are really concerned about a class of pesticide called neonicotinoids, many of which are highly toxic to honeybees. And most corn seed in the US is treated with neonics. There's 3 different ways that bees can become exposed to neonics on corn. First, during planting, a lot of dust is created, and that dust can settle onto bees themselves and onto wildflowers, causing acute mortality events. Second, the pesticides leach into the soil and can be taken up by wildflowers, which the bees later visit. And third, because neonics are systemic pesticides, they're taken up by all parts of a plant, including its pollen. So bees that are foraging on corn pollen could also be bringing pesticides back into their hive. And beekeepers were really concerned about this third route of exposure. They wondered, if my hives are near a cornfield, are my bees bringing in a lot of corn pollen? And if so, are they also bringing pesticides back into the hive? So our research focused on this third route of exposure. And to do so, we partnered with beekeepers across central and western New York, where most corn and honey is produced. And we sampled 49 hives across this region, and in each hive, we collected fresh pollen out of the hive and screened it for a number of pesticides, including neonics. On this map, we have the white points representing the 49 hives that we sampled, and they were within 400 meters of cornfields, which are in orange, and then yellow represents other crops. So we asked 2 questions. First, how much corn pollen are bees collecting? And second, how much pesticide risk are they experiencing? So first, we found that bees were not collecting a lot of corn pollen. 61% of samples did not have any corn pollen, and all samples were composed of less than 5% corn pollen. Second, we assess risks using the pollen hazard quotient. And this just incorporates information on exposure, so the concentration of pesticides within the hive and the toxicity of those pesticides. And we found that pesticide risk was always low, less than 3%, and this was not related to how much corn pollen was in the hive. We also looked at the landscape around these hives, and we predicted that hives surrounded by more corn would have higher pesticide risk than hives surrounded by less corn. But we found no relationship between pesticide risk and the amount of corn around a hive. So overall, first we found that bees were not collecting a lot of corn. Rather, they were collecting other things in the landscape, other crops or wildflowers. And second, that pesticide risk was generally low, which was really encouraging. So in 2016, we wanted to trace pesticide risk through time. One beekeeper volunteered to use pollen traps to collect pollen weekly from 4 hives in one apiary that was really close to a cornfield. And we found that in August 1st, bees were actually collecting a lot of corn pollen. About 40 to 60% of the pollen coming in was corn pollen. However, pesticide risk was quite low August 1st, almost 0, and only peaked about 2 weeks later in one hive. So overall, we found that pesticide risk was quite low in both years and was not associated with how much corn pollen bees were bringing into a hive or how much corn was surrounding that hive. This, of course, was only conducted during the corn flowering period, so that third route of exposure. And we still need to understand those 2 other routes of exposure, dust settling on bees and wildflowers and leaching into the soil and being taken up by wildflowers surrounding fields.